Positron emission tomography is a very big term. Maybe another different title which would have worked would be cerebral energy metabolism, because that's at the core what we're going to study. The brain essentially is a biochemical machinery. And of course, this machinery needs some energy. It needs energy such that it allows us to have cognitive function and ultimately to have behavior. Some numbers. The brain consists of 86 billion neurons, and these neurons are energy hungry. In fact, they consume, or they occupy 20% of the whole body's energy consumption, which is remarkably high given how small the brain is compared to the remaining body. So it's a very energy hungry organ. And at resting state in a healthy brain, so when we're awake and doing nothing special, this consumption, this 20%, are roughly constant. So there's a myth that hard thinking makes us consume more energy, but that myth is not true. Why is this relevant for clinical research? Well, we can study the energy metabolism of the brain. In a healthy brain, for example, there will be specific patterns, there will be specific energy consumption in certain brain regions. However, if you look at pathologies, for example, Alzheimer's disease or Lewy body dementia, these patterns can be altered and the energy consumption can be altered. So from a clinical perspective, it's important to use PET scans in order to understand these diseases. Just as a side fact, typically in clinical routine, PET scans are used for cancer diagnosis, but my presentation is not about this part. So another example, this is less clinical, but more relevant for basic research, for understanding how the brain functions, is the energy consumption of the brain in different states of consciousness. As an example, on the top panel, you see an awake brain. In the bottom panel, from this 2001 study, you see brain in non-REM sleep. Non-REM sleep is one of the phases of sleep. And in this study, they have found that in non-REM sleep, the energy consumption is reduced by around 15%. So relevant for clinical research, relevant for basic research. How do we assess this energy metabolism? Well, the answer is, that was my title, is 18-FDG PET. So PET is one method to, is an imaging technique, and 18-FDG is one particular imaging technique. PET stands for positron emission tomography, that is based on a physical phenomenon known as electron-positron annihilation. That's a reaction between matter and antimatter. The procedure is the following. First, the subject gets a radioactive tracer injected. It's a low dose, but the scans should not be done too often still. This radioactive decay is designed in such a way that it produces positrons. That's the antimatter counterpart of electrons. And when positrons and electrons meet, they perform so-called annihilation. So they disappear in the form of energy that is emitted in photons. So this annihilation between positron and electron emits two photons. And what's important about this emission is that those photons are emitted in exactly opposite directions. This means if we can measure those photons in a detector in those opposite directions, we can trace back where the annihilation came from. In other words, where the tracer is. And 18-FDG PET in particular, there's different types of PET, but I'm talking about 18-FDG PET today. It's a tracer which is essentially looks like a glucose molecule, but something was manipulated in it such that it's radioactive and produces those positrons. So with 18-FDG PET scans, we have an imaging technique which allows us to know where glucose goes in the human body and also in the brain. So, as I said, there's clinical relevance for this. Of course, there's also, for us who use a lot of modeling to understand the brain, there's relevance for the modeling, because what the virtual brain does, what we do with digital brain twins, is essentially simulating raw neural activity. That might be, for example, some macroscopic measure like mean membrane potential of a population of neurons or the firing rate of a population of neurons. Those are things that we cannot measure empirically. And if you fiddle around long enough, a simulation can produce any type of dynamics, any types of activity patterns. So somehow we need to make sure that this neural activity is meaningful. By meaningful, I mean it must somehow relate to empirical data. The issue is we cannot measure raw neural activity in a healthy brain. Like we would need, it would need some invasive method. Instead, what we measure on the empirical side for brain activity is EEG, BOLT, or imaging. You heard about this, I guess, in the past weeks. And now the question is, can we also study PET signals using the virtual brain? Because if we add another modality to this picture, it will allow us to inform our models better, to improve the predictive power, to make sure that our simulations are meaningful. So what can we do? We need to develop a new model for this. And the model that we develop is a PET model, but at its core, it's a neurogliovascular model. There's 3 words in this. There's neuron, there's vascular, referring to blood, and there's glio, regarding to glia cells. Glia cells are non-neuron brain cells that are responsible for structural support in the brain, but also for metabolic regulation. And metabolic is where the energy metabolism comes into play in this picture. So let us start from scratch. We don't have a model, and we want to arrive at something like a PET image that you see on the right side. What we have on the left side is a small scale, which is, in these virtual brain simulations, neural activity. And somehow we need to bridge these scales from neural activity to PET scan signals that are measured in the scanner. So for this, we need to answer the question, since 18-FDG is similar to glucose and glucose goes where energy is needed, why is energy needed in the brain? Well, neural activity requires energy, and the reason is the spiking activity of neurons. So typically a neuron is known for its spiking behavior, so for the emission of an action potential, and that works through ions and through ionic gradients. So when a neuron receives input that is strong enough, it might trigger an action potential. During the rise of the action potential, sodium goes out of the cell. At the decay of the action potential, potassium goes out of the cell. That's the repolarization phase. And then the cell goes back to resting state. However, this procedure changes the ionic gradients. And in order to bring the ionic gradients back to the initial state, we need energy because the ions need to move against the gradients. What is responsible for this reset is the sodium-potassium pump that brings sodium out, potassium in, and this activity requires ATP. That's the basic energy unit for us. So when the neuron spikes, energy is consumed, so the ATP concentration changes. And in order to make sure that there's always enough ATP, the cell somehow needs to send out a hunger signal, a demand signal, asking for more energy. And it is this hunger signal which leads to the uptake of glucose. So let's put this together in a schematic way. This is a very basic neuron with a soma and some neural activity. There's some ATP in the cell. Neural activity will trigger ATP consumption. The glia cells will send out a hunger signal, which leads to the influx of glucose into the cell. So this is where the glucose uptake happens, and that leads to ATP increase to even out the ATP levels. Now, the thing with 18-FDG PET is that 18-FDG and glucose, they share the same enzyme that is responsible for trapping the molecule inside the cell. This process is called phosphorylation, and the enzyme that is responsible for this is hexokinase. So as soon as phosphorylation of glucose increases, phosphorylation of FDG will also increase, and it will be trapped inside the cell. So with the glucose uptake, we will get also FDG uptake. That is ultimately what gives us the PET signal. So this is the block of our neurogliovascular and PET model. This is so far just schematic. In order to perform brain network simulations with such an approach, we need a model. We found this model in the literature from 2012. This is a very elaborate model, basically describing on a microscopic scale how this energy household works. It's an 89-dimensional system. You can imagine that we cannot simulate 89 equations per neuron, and we want to simulate a whole brain. That's not possible. So we need to reduce the complexity by a lot. We need something that is tractable in order to be able to use neural mass models for the simulations. So we simplify the whole thing. We know that the PET signal is proportional to the glucose uptake, and that is proportional to the ATP concentration. So we just need to find a description for the ATP concentration based on some very, very basic assumptions. Those assumptions are the ones I mentioned. At each spike, ATP is consumed. At the same time, production is increased to increase the supply. And at rest, when there's no spike in activity, ATP goes back to the baseline. We can put these three assumptions in a model, in a mathematical model. So there's these two differential equations. The first one describes the ATP concentration in a given region. Now, this is region-wise. This is already on neural mass level. And the second equation, F, describes the production, so the hunger signal. What happens is, on spike, A decreases, hunger signal goes up. And the fact that this is dependent on the spiking activity is captured by this R here. That is the firing rates, for example, or the some neural activity level. So if neural activity goes up, R goes up, and then the A and F follow some dynamics. Okay, so far so good. Now we have a PET model, very basic, very simplified based on the biology. And we put this neurogliovascular and PET model in a separate block. Now, how do we get the neural activity that is driving this model? As I said, we cannot measure it directly in imaging. Instead, we use virtual brain simulations. So simulating neural activity in the virtual brain. This is now a zoom into this left part of the pipeline. We generated in the virtual brain. What are the ingredients for a whole brain simulation? The first ingredient is a neural mass model, a model that does not capture the dynamics of individual neurons. We don't care about the individual neurons. We want a model that captures the emergent macroscopic dynamics of whole populations of neurons. So mean membrane potentials, mean synaptic potentials, or mean firing rates in this case. Second ingredient is structural connectivity. That is something that is obtained through DTI and through tractography. Basically, it tells us between pairs of regions how many fiber tracts are there. How are they physically coupled, actually? How strongly are they coupled? And this is basically what we need for a whole brain simulation in order to produce some neural activity time series. So, as you might know, whole brain simulations have parameters. Depending on those parameters, the outcome might be completely useless. So we need somehow data in order to verify that this neural activity that we're producing is meaningful. For this, we used fMRI data. So we have, we have fMRI time series from which we can compute functional connectivity matrices. These are these things that you see at the bottom. They tell us how strongly the signals of two brain regions are correlated, so how synchronized they are. The brighter the pixel, the more synchronized two brain regions are. We can compute such a functional connectivity matrix on the empirical data, and on the simulation data. On the simulation side, we can take the neural activity of the simulation, feed it into a Bose model, into an fMRI model, in order to compute a simulated functional connectivity matrix. This is the model we can use here. It's called, I will not go into the details. I will just tell you what's important about this. It's the so-called generic 2D oscillator. It has two equations. What's important is that it has typical properties of neuronal dynamics. That is, for example, multistability, oscillatory behavior, and dynamics on multiple timescales. And for those of you familiar with the terminology, the generic 2D oscillator is basically a generalization of the Fitz-Yanagumo type models. Okay, this is the data set we used. It's a data set of 27 healthy subjects from the Monash University. It contains remarkable data because it consists of Bolt time series as well as FDG PET time series. So they were both measured at the same time. There's not many places having such setups that can measure both at the same time. So we use this data because we will need both data parts, the fMRI part and also the PET part later. Let's focus on the fMRI part for now. You heard, I think, in one of the sessions already about optimization, about fitting. The method I use here is very basic. It's a grid search. So I take two of the parameters of the model that I don't know, and I build a grid out of these two parameters. That's what you see on the top left. For each point of that grid, I simulate the whole brain. I compute a bold time series. I compute a functional connectivity matrix for each of these pixels. This is the functional connectivity matrix that you see at the bottom. The one at the top is the one from the empirical data. And you see the brightness of the pixel tells us how close these two matrices are to each other. So the brighter the pixel, the more close are the fMRI characteristics of the simulations as compared to the empirical data. So I picked already the best parameter here. So the functional connectivity matrices that you see here are already the best fit. But just to illustrate how badly simulations can go, I will swipe a little bit randomly through this space. And you see some of these matrices are completely noisy. Some of them are fully synchronized. Some of them have a very large separation of the hemispheres. But if we pick the brightest pixel, we get a reasonable agreement. Okay, so now we have a whole brain simulation that is tuned such that it produces characteristics of fMRI data. This is our TVB neural activity and fMRI simulation. Now we put back the PET model into this picture because what the PET model needed, the 18FDG PET model needed, was some neural activity time series. And we have this now. So we can take this neural activity time series and feed it into our PET model. The issue is the PET model itself had three parameters. Those are the green ones. This A decrease, tau F, and F increase. So we need the PET data of the data set in order to choose those parameters to make the PET model reproduce the PET data, the empirical PET data, as good as possible. So essentially, we take the empirical PET time series from the data set. We take the simulated PET time series with random parameters. Then we compute the so-called static PET image. That basically tells us how much PET signal is in each brain region. It's one value per brain region. And we can compare these two. So we compare the empirical static PET with the empirical static PET. The simulated static PET with the empirical static PET. This is a static PET image on the right that you see here. And then we use some iterative method to tune those three parameters in a way that will improve the agreement between empirical and simulated static PET. So this is an older version of this approach that we had. We used a so-called genetic algorithm. I don't have the time to go into detail, but essentially it's an iterative procedure that, as you see here, the fitness, so the fit between empirical and simulated data increases over the iterations. Then we take the last iteration of this optimization. We take these three parameters, and we can compute the static PET image of all the subjects. The optimization was performed only on 3 subjects, but now we can compute them with the optimized parameters on all the subjects and compare the empirical data with the simulated data. And this is the result. So on the left, you see the empirical one. On the right, you see the simulated data, which have a correlation of 0.56 as a group average. If you look at individual subjects, the correlations go up to 0.7. So it's not a perfect match. We're not simulating the perfect brain of those subjects here, but the correlations are quite high. And they are surprisingly high given what we're trying to do here, because if you think about it, what we did is we infer neural activity from fMRI data. We take this neural activity and feed it into a PET model and get PET data out. So we infer PET from bold. Okay. I want to go back to sleep. That is something I mentioned in the beginning. It's one of the possible applications of such a model. As I mentioned, in the non-REM sleep brain, the energy consumption of the brain is 15% lower. I was talking now all the time about an FDG PET model, but what we measure with 18FDG overall is glucose uptake. So it's the energy landscape of the brain. Can we somehow reproduce this in a simulation? Well, we thought it's a little bit difficult to simulate an actual awake brain or an actual non-REM sleep brain. What we did instead is to look at characteristics of a brain in awake state and in non-REM sleep. One of those characteristics is the relationship to brain oscillations. Typically, an awake, healthy brain, if you measure the EEG, you will find mainly with a high power alpha band activity. That is the band from 8 to 12 hertz. Whereas in non-REM sleep, the activity is in the delta band from 2 to 4 hertz. And we can exploit this fact by making simulations that are not like proper awake and non-REM sleep brains, but instead have these characteristics. So we need a simulation that has alpha band activity. We need a simulation that has delta band activity. For this, we use the Janssen-Ritt model. It has 6 equations. Don't look at them. It's awfully complicated. I will explain it in a different way. What is important to know about this is it models the cortical column, that is, so it contains of multiple neural populations, and it gives rise to complex dynamics, in particular those frequency bands that we need. Instead of going through the equations, I will show you what the model can do. So these are simulations. One thing to mention is each neural mass model has one parameter that is common to all neural mass models. That is the external input. So how much activity does the neural mass model receive from other regions? In a whole brain simulation, this external input is something that changes in time because activity of the brain regions is changing in time. But if we look at the model in isolation, this external input can be constant. So in this case, I chose an external input of 0.05 for the Janssen-Ritt model. And I can just run some simulations and see what happens. So this is a single region. We see one of the simulations starts high and goes to some flatline baseline activity. One of them oscillates and then goes down. One of them stays up a little bit longer and then decays. So there's some interesting dynamics going on. And if you would count the oscillations here, you would realize this is alpha band activity from 8 to 12 hertz. And now if we change this external input parameter, we observe something completely different, something that looks like delta band activity. But depending on where we start, we also get the alpha band activity. So this is something we know as bistability, multistability. There's 2 attractors in the system. Depending on where we start, we go to one or the other. So as I said before, when simulating an actual whole brain with many regions, this parameter, which is 0.12 right now, is not constant anymore. But each brain region will receive an I external that is time dependent and different depending on the region, depending on how much input it receives. So in order to show this in a nice plot, we need 3 axes. We need this y0 and y3 that we saw before on the left panel as a time series, but we also need the I external axis. Each of these dots represents one brain region. You see the I external of those is not constant. And in fact, if I run the simulations, they wildly oscillate around. These circles that they make are in the alpha range. And this is like, it looks cool, but what do we make out of this? So by the way, this was the wake-like simulation. So this is fitted to ball data again. Well, what we make out of this is we find out more about this by performing so-called bifurcation analysis. It's a complicated term, but I will explain it very briefly. So what you see here is a bifurcation diagram. It shows the attractors of the system. It shows what possible dynamics are available at a given value of this external current. And you see these structures. This white line here is a so-called fixed point. This corresponds to this flat line we saw before. The orange surfaces correspond to oscillatory behavior, periodic motion. This elongated football corresponds to the alpha band oscillations. And this narrow band here corresponds to the delta band oscillations. And the cool thing about this is right now there's this brain network simulation, the dots, which are moving around, but the bifurcation diagram, the orange stuff, we can compute it beforehand without knowing anything about the brain network simulation. So we can predict using this bifurcation diagram how the system will behave. And if I run the awake-like simulation plotted on the same bifurcation diagram, we see that, yes, they are on the alpha limit cycle. Okay. And now we have an awake-like simulation, which is producing alpha-like oscillations. How do we get it to produce delta band oscillations? Well, we somehow need to bring the system further left, such that the regions, the individual brain regions here, can reach the delta band attractor. And we do this by lowering the global coupling across brain regions, because this will lower the input they receive. It will shift them to lower currents. So this is now the simulation of the non-REM sleep-like state, which is oscillating on the delta band limit cycle. Okay, so now we have the fitted simulation. So fitted on resting state, it produces alpha band oscillation. And we have the simulation with lower global coupling that produces delta band oscillations. We can take our PET model now, feed these neural activity signals into the PET model and measure the PET response that basically tells us how the glucose uptake is in certain brain regions. And what we find, remarkably, even though this model is very simple, is a reduction of the energy consumption from 100% to 85%. Yeah, 85%. So reduction by 15%, analogous to what we found in the empirical data. Okay, that's it. That's basically an illustration how to tackle a complicated problem like developing a PET model from scratch. And I think it covered many of the key concepts of the last weeks, like neural mass models, whole brain simulations, fitting, and some others. I have some take-home messages. So the bifurcation analysis I've shown is quite mathematical. It's very technical, but it's very important. In my opinion, it's a very important tool in order to get a deeper understanding of some model's behavior. It also facilitates its exploration, because if you don't know the structure, you wouldn't know what to change in order to make the brain produce delta band oscillations. So knowing this bifurcation structure, and there's tools which you can use to compute those, helps us in general in brain network modeling. Second take-home message, we have seen this very complicated model with 89 equations. Of course, if you're interested in all the details, we need to do this. But in this case, it was sufficient to use 2 equations and a neural mass setup. So second take-home message, for complex problems, we need to choose an appropriate level of biological detail. That is not always easy, because you can always add more effects or remove effects. You will have to find some middle ground here. And third take-home message is regarding basically this multimodal modeling approach. As I said, we have already the ways we can produce already EEG from the virtual brain. We can produce fMRI or MEG, and now we can also produce PET. And the more modalities we have, the better we can make the models. The more trustworthy, the more trust we can put in the models. So take-home message 3 is multimodal approach allows better model validation and improves predictive power. And with this, I finish. Thanks for your attention. I'm happy to take your questions.
Hey everyone. Today I'm going to speak about using a brain network model to understand the pharmacodynamics of ketamine. And firstly, I will give you a brief history of ketamine and what it is medically used for. And then I want to explain to you how we can use a brain network model to get more insights into the mechanism of action. So, ketamine was firstly approved as an anesthetic in the 1970s by the FDA, and it is on the essential medicine list of the World Health Organization. And it is a very important medicine in emergency use because it preserves the hemodynamic and airway reflexes. And while using ketamine as an anesthetic, patients reported vivid dreams and dissociations. And this was first seen as noise and usually masked with given benzodiazepines. And ketamine is an NMDA receptor antagonist and not a GABAergic anesthetic like the most common anesthetics like propofol. And this one difference really shapes its whole clinical and psychoactive profile. So ketamine has a graded dose spectrum, so you can give it in low doses and high doses. And after giving it as an anesthetic, after that time, people experimented with different doses. And in low doses, it has mild analgesia, which means it is pain relieving, and it's also used in chronic pain patients. In low doses, it also induces a psychological state of dissociation, which is an altered state of conscious where individuals feel detached from their bodies, surroundings, and sense of self. Also described as depersonalization and derealization, or also sometimes called psychotomimetics. So it seems that it induces psychosis. And in high doses, it induces anesthesia. And in this period of time, they found out that ketamine is not, the dosing of ketamine is not a monotonic deepening of anesthesia like with other anesthetics like propofol, but really the dose response passes through really distinct phenomenological regimes. So after finding that out, there started a period where they did a systematic subanesthetic characterization with lower doses of ketamine. And in this period of time, they started observing that ketamine induces a state that models schizophrenia or psychosis because it really captures the positive and negative symptoms of schizophrenia. The positive symptoms include hallucination and delusions, and the negative symptoms are, for example, anhedonia. This also then motivated the NMDR hypofunction hypothesis of schizophrenia. This hypothesis states that there is a impairment in the NMDA functioning at GABAergic interneurons. So you have reduced functioning of these NMDA receptors, especially so you have an impaired like glutamate signaling, particularly in the cortex and in the hippocampus. And then in 1990s, there were the first controlled dosing of human volunteers with subanesthetic doses to investigate these dissociative states. Ketamine as an antidepressant. So in the 1990s, the glutamate system became more and more implicated in mood disorders. And there was the first rodent study showing that NMDR antagonists exhibit antidepressant effects. And then 10 years later, there was the first human studies, a study showing that a single subanesthetic infusion produces rapid antidepressant effects in humans. And then 19 years later, the FDA approved the intranasal esketamine for the treatment of treatment-resistant depression. And currently, also in Europe, ketamine is used off-label to treat depression, also with therapy, and it's currently also investigated for a multitude of mental health disorders. So next to the behavioral effects, we can also observe physiological effects that are dose dependent, and they include changes in the glutamatergic neurotransmission and also electrophysiological effects. So it again ranges from low dose to high doses. And in the subanesthetic low doses, we usually observe a pyramidal firing rate increase, a decrease in alpha power in the EEG, and also a decrease in theta and delta low frequency power. And in the anesthetic dose, we see a reduced pyramidal firing rate, a decrease in alpha power, and an increase in theta and delta power. And this is later important because this is what we want to reproduce with our brain network model. And of course, ketamine also induces a lot more like physiological effects, but we will focus on these ones because this is what we can really reproduce with the brain network model. And we're more looking at EEG and also the pyramidal firing rate and not at fMRI in our case. So, ketamine's main mechanism of action is the NMDR antagonism. So this is the NMDA receptor, which is an excitatory glutamate channel. So I hope you can see my mouse. So this is a membrane. This is the intracellular space. This is the extracellular space. And here is the NMDA channel, which consists of 4 subunits. And it has glutamate and glycine or the serine has to be bound both so the channel can open. And it's an ion channel and it lets the positively charged ions of sodium and calcium through. But it also has this magnesium block that you see in the open pore, which also blocks the channel. So this magnesium has to be removed before the channel can open, and this is usually done by depolarization. And ketamine is an open channel blocker, so it blocks deep within the channel. So the magnesium block also has to be removed before ketamine can bind the channel and block it. So there are different theories about how ketamine induces these distinct dose-dependent effects, and they are usually derived from like rodent research. And I will introduce the most prominent ones, and they are usually so. This is a synaptic model. This is a pyramidal cell that you see here. This is the pre-synapse. This is the post-synapse. A depolarization comes from here. Here you can see the synaptic vesicles with the neurotransmitters in them. And on this side, you see an inhibitory interneuron, which inhibits the pyramidal cell via GABA. And the NMDA receptors sit, for example, here in the excitatory neurotransmission on this pyramidal cell postsynaptically. But we also find NMDA receptors on these inhibitory interneurons that then excite the inhibitory interneuron, leading to inhibition. So ketamine now, depending on where ketamine binds on which of these neuron types, it has different effects. So if ketamine binds on this inhibitory interneuron here and blocks the NMDA receptor, we get less inhibition. Therefore, we get increased neurotransmission along this pyramidal cell. So this usually happens in low doses, and this is called the disinhibition theory. And this is one of the most influential theories about ketamine's mechanism of action. So it just states that ketamine causes a disinhibition along this inhibitory interneurons here. And ketamine can, of course, also bind these receptors and block them. So you get decreased excitatory neurotransmission. And this is supposed to happen more in the anesthetic dose. And this is happening in the low dose, that it preferentially binds NMDRs on inhibitory interneurons. And in higher doses, also the NMDA receptors on these excitatory pyramidal cells are more involved. And now we want to bridge the gap between these like molecular level effects that we see and the physiological effects that we are observing in empirical research. And we're doing this by using a brain network model. So as Halgurd said in his presentation, for this we're using first a structure connectome to create a graph of the brain, where each of these like nodes is represented by a population model. In this case, it's also the Janssen-Ritt neuromass model, just as Halgurd said. And this model, so this is a schematic representation of the model. The model is a biophysical model, so it has like properties and parameters that are grounded in biology. So each node here then consists of three subpopulations. This is the pyramidal cell. This is the excitatory interneuron. This is the inhibitory interneuron, and they are locally coupled by these local coupling parameters that you can see here. And now what we want is that the mechanism that I told you before with these NMDA receptors, we want to translate this on a population level into this model. And we're doing this by targeting these local coupling parameters that are an excitatory drive, but they are lumping together AMPA and NMDRs, but still they are related to the NMDR-ergic activity. As a first process, we need to fit the model, just as Halgurd said. So in our case, we fit the global coupling, which is the strength of how strongly the nodes are connected to each other. And we also fit the conduction speed. And we are fitting first with a bold FC, so a functional connectivity, just so we see that our model can create a biologically plausible functional connectivity. This is our simulated FC, and this is the empirical FC. So now we have model parameters where we can just run a simulation. So what Halgurd showed you in a 3D structure, this is in a 2D structure, the bifurcation diagram of the Janssen grid. It's just basically on the left here, it's the bifurcation diagram with the tractor landscape. And on the right, you see the possible frequencies of the oscillations. And again, we have the external input, just as Halgurd said. It's how much input each node gets from the and in the case of a single node, this is just a stable number. And what you can see here in this area, the model exhibits an alpha frequency. And in this area here, the model exhibits low frequency oscillations. So as a next step, we are implementing ketamine through 3 parameters, the local coupling parameters. And as a first step, you need to do like a parameter exploration just to see how sensitive your parameters are. This is now a lot, so let's look at one panel. So as I told you before, we want to investigate the firing rate and some electrophysiological properties. So on the y-axis, we find the inhibitory coupling. And on the x-axis, we find excitatory coupling, and each panel is the other excitatory coupling. And then we can just see, okay, in which region does the model exhibit a reasonable, biologically plausible firing rate, and when does the firing rate drop to 0, for example. And this we can then see for different values of the excitatory coupling. And we're doing this for also the alpha power to see in which range the model exhibits alpha oscillations. And we also can do that with the theta and delta power. And what you can basically see for all of them is that the inhibitory coupling is a bit more sensitive to change than the excitatory coupling. And this will be important a bit later. So now we can start to test like hypotheses and start to implement ketamine into the local coupling parameters. And we do this by using sigmoidal transfer functions that translate the ketamine that ranges here between 0 and 1 onto these local coupling parameters. They are normalized in this case, so we can have them on the same axis. So it just means they're divided by their maximum value. And as a first step, we want to see what happens if ketamine impacts all the NMDR receptors on all neuron types homogeneously. That's why we have the same sigmoid for all the local coupling parameters. This dotted line is the midpoint of the sigmoid, which states the point where ketamine acts the strongest on the local coupling parameters. So they all are changed the same. Then we can look into the excitation inhibition ratio of these local coupling parameters over the course of ketamine dosing. And here you can see it just stays at 1 because we are changing them basically all the same. Then we can look into, okay, how does the power behave over like ketamine? And we can see, okay, alpha power just goes down. This is already good because this is what we want to find, consistent alpha decline. But we also see that for theta and delta in blue and in purple, they all just go down, which is not what we expect because we want them to go down and then up in an anesthetic regime. And when we look at the firing rate, it's also not really the behavior that we want to see, because what we want to see is that the firing rate first increases with higher ketamine, and then in anesthetic doses of ketamine decreases again, but here it just increases. So then as a next step, we were like, okay, let ketamine impact the NMDRs on the neuron types heterogeneously. As I told you before, it seemed that the inhibitory coupling parameter is more sensitive and has a smaller range of biologically plausible firing rate and also alpha and theta power. So that's why I decided to change it a bit less percentage-wise than the excitatory coupling parameters. And the inhibitory coupling parameter you can see in pink. So it's changed a bit less than the others, but they still have the same midpoint. So the ketamine still impacts them at the same dosage, the strongest. And then the excitation inhibition ratio now decreases over the whole dosing regime. And now what we find in the power is, okay, like alpha power in green still decreases across the ketamine dose, which is already good. We had that before. But now we also find that in an anesthetic regime, which we would set at 0.5 in this case, then, oh, theta and delta already increase. So that's good. That's already better than what we had before. But when we look at the firing rate just decreases. So still not really a realistic firing rate behavior that we're looking for. So as a next step, I then additionally to decreasing the inhibitory coupling a bit less, I also changed the midpoints of the sigmoids so that the strongest impact of ketamine is at a way earlier dose for ketamine for the inhibitory coupling that you see the pink dotted line here. And for the excitatory coupling parameters, it's at a higher ketamine dose. So now the excitation inhibition ratio first increases and then decreases over the ketamine dose. And what we find then is again a decrease of alpha across the dosing. Then we find a decrease in theta and an increase in anesthetic doses for theta and delta. So that's perfect. That's exactly what we wanted to find. And now when we look at the firing rate, we also find a firing rate behavior that we are seeing in empirical research, which is an increase in firing rate and then a decrease in anesthetic regimes. So, as a summary, ketamine has a dose response with distinct phenomenological regimes and physiological effects. And a brain network model can help us bridge the scales. So we can turn a receptor level hypothesis into a testable predictions about firing rate and spectral changes. And our model shows here that only a heterogeneous antagonism is able to reproduce the physiological changes that we see in empirical research. So thank you for your attention. Was it from my side, and I'm open for your questions.
Okay, thank you, Jenny, for your talk about ketamine. And actually, we will continue talking about clinical drugs. In this case, more about drug repurposing. So getting from drugs that are being used in a different setting for treating diseases that originally were treated by other medications, pretty similar to what we've heard about ketamine just a few moments ago, which was originally used as an anesthetic and later also used for treating depression. There are many cases in medicine where drugs that were originally developed for a certain use case were later repurposed for some other disease. And what we try to do in the research I'm going to present here is investigating drugs that are already in use and that are already given to patients for certain use cases, and screening the literature and knowledge that we have on these drugs for finding drugs that are applicable for being repurposed for Alzheimer's. And since this is the summer school of virtual brain simulation, of course, we will also talk about that and talk about how to make use of virtual brain simulations to augment or even to enhance drug repurposing. And actually, there were two sessions in the summer school already that were associated with that topic. One of them was by Leon Martin. I think it was the second session in the summer school about the virtual brain ontology. And the virtual brain ontology concepts are very much related to what I'm going to talk about today. So you'll see a couple of plots that were from the paper that Leon Martin presented. And I know that you've also heard quite a lot about Alzheimer's disease in the summer school, in particular by Leon Stefanowski, who's also associated with the research I'm going to present. And as you may remember from the TVBO virtual brain ontology presentation by Leon Martin, we do have many different variables in our brain simulation network or brain simulation framework of the virtual brain. And some of them are more theoretical than others. So we do have some variables in our models that are very much based on biological parameters. And then there are others, for example, the models that model epilepsy, that are more mathematical and less of a surrogate of some specific biological marker or channel or something like that. And what you can see on this plot, and we will return to this graphic at the end of this talk, is that what we're trying to build with the virtual brain ontology and the approach of virtual brain drug repurposing is getting a connection between a disease mechanism. I hope you can see my mouse. The biological processes that are associated with the diseases, and from there to the mathematics that are actually the surrogates of the biological processes that drive the disease. So before we go into the details here, we first need to understand that Alzheimer's is a pretty complex disease, and there is no pathological mechanism that's widely accepted as the driving mechanism of Alzheimer's. Of course, there are many hypotheses, and there are even drugs that are given to patients that are diagnosed with Alzheimer's. But at the moment, we don't have a single understanding of what drives Alzheimer's disease, and we don't have an understanding of what specific biological process drives it. But what we do have is a list and some understanding of biological processes that are associated with the mechanisms that somehow lead to Alzheimer's. So there are hypotheses about tauopathies. There are hypotheses about neuroinflammation and their relation to tauopathies. And we do have some understanding of what biological processes are upregulated within patients that suffer from Alzheimer's disease. And we also have some understanding of what drugs have an impact on these specific biological processes that are at least associated with Alzheimer's. Perhaps they are not the single cause of Alzheimer's, but we do have an understanding of connected biological processes. And of course, there's a lot of knowledge and a huge and fastly growing literature, especially written within natural language, on pharmacodynamics and these biological processes. And it's pretty hard for an individual researcher to go through all that manually, through all the drugs that are on the market that somehow have an impact on the processes that drive Alzheimer's disease. So what we try to do is applying the technologies that have been introduced in the last couple of years within large language model research that allow us to process natural language and knowledge written in natural language, and to combine that with some structure or knowledge structure that allows us to later mathematically model what we see written in natural language. And this is the first step of that research pipeline, that we first need to get an understanding of what biological processes are actually driving Alzheimer's disease. And there are different ways to categorize biology. And one of them is, as Leon Martin already introduced you to that concept a couple of days ago, are ontologies. So ontologies are basically highly structured, highly hierarchical organizations of knowledge and of terminology, which allows to organize and structure knowledge, but also to make knowledge interoperable. And for biological processes, there is an ontology that is called gene ontology. And that name is sometimes a bit misleading because it's called gene ontology, but it also applies to biological processes. So the gene ontology is a ontology that's made for structuring biological processes in this highly hierarchical mechanism. And we'll have a look at the gene ontology later. But for now, it's just important to know that there are certain concepts, certain biological processes that are encoded in the gene ontology. And the good thing is that often when people do research on biological mechanisms, they can encode their research in the gene ontology to make sure that when they talk about a certain term, that everyone that uses that term talks about the exact same thing. And there's a data set that was introduced by ROQCL that did exactly that, having a look at the biological processes that are associated with Alzheimer's disease. It's 1,778 in total. And there's also something that's called the drug bank. And the drug bank is a big database that contains pretty much all drugs that are in use in the clinics, but also that were only remained in preclinical stages or in RCTs or sometimes even within just lab studies. So we got the drug bank, and you can see that here, that consists of 16,500 different drugs. And I mentioned before the gene ontology standardizes biology, and ontologies allow us to become interoperable. So if one ontology is connected to another ontology, you can connect different domains of knowledge that otherwise could not be connected in a standardized way. And the gene ontology, in its way, how it encodes biological terms, is interoperable with the drug bank that also has gene ontology terms associated. Unfortunately, they don't encode the terms directly, but there's a way of connecting them since they share one knowledge base. So what we did is having a look at the drug bank and seeing there are 16,500 drugs, and we know that there are 1,778 biological processes that are somehow associated with Alzheimer's. If we want to find and identify drugs that are interesting for drug repurposing, we can have a look what drugs out of this large drug bank database actually share biological pathways with what's driving Alzheimer's. And that's about 6,000 of them. So these 6,000 drugs are theoretical candidates for drug repurposing within Alzheimer's disease, as they at least have an impact. We don't know yet if it's a good impact, if it enhances Alzheimer's disease or something, but we do know they somehow have an impact on what is associated with Alzheimer's disease. Now to get into more detail, for example, understanding if the biological process is just associated or if it's regulated up or down or something like that, you need to go through pharmacodynamics. So this is nothing that would be explained within the gene ontology. That's something you need to have an actual understanding of how the drug behaves within biological organisms. And this is not as strictly organized as the biological processes themselves within ontologies. But since we do have large language models at hand that are actually able to get at least some basic understanding based on natural language, if, for example, a process is regulated up or down, or if it's not even applicable within the central nervous system, as it can't cross the blood-brain barrier, and all these aspects that actually play a role for drug repurposing. This is something we believe a large language model is actually very much able to comprehend within guardrails, of course. And so the first step that we did after filtering these drugs that are somehow associated with Alzheimer's disease driving processes is to provide them to a locally hosted large language model, but not just the drug itself, but also information about its pharmacodynamics, its mechanism of action, clinical trials, and all that kind of knowledge that is actually only written within natural language that otherwise would be missed if we wouldn't include it. And then we asked the large language model to provide a rating for these drug repurposing candidates, to have some kind of pre-selection, not to let the LLM decide what drugs are actual candidates, but to have a pre-selection of interesting candidates to look at, to not having to go through these 6,000 drugs and all the data associated with them manually, but to have some kind of a pre-review of applicable knowledge. And this is what that looks like if you run that through a graph database that contains the drugs, the biological processes, and the disease that's affected. You've got, for example, reproducible here, which is a kinase inhibitor, usually used for treating certain kinds of gastrointestinal cancer. But it also has an impact on a wide range of biological processes that are associated with Alzheimer's disease. So in theory, if a drug such as this kinase inhibitor would inhibit one of these biological processes that drive Alzheimer's disease, it might actually be a candidate for being given to Alzheimer's disease patients or people that are at risk of it, as it could inhibit that specific disease-driving biological process. And as you know, there are many reasons to be very careful about what comes out of throwing data into large language models. And one of them is certainly is built-in randomness in their answers. I mean, their likelihood predictors. So one of the major caveats one has to have when making use of large language models for predicting something is to compare how does it perform over different iterations. So how much of the actual results are associated with randomness and how much of it has actually something to do with that large language model reasoning, which doesn't mean the reasoning is correct, but at least one has to differentiate between randomness within the model and its own reasoning. So we did not run those iterations once, but 10 times to have a comparison between the performance of specific drugs across iterations. And this is actually what you can see here on this plot. So what that plot shows is each individual drug compared across each iteration on how on its performance. And actually, what we see from that is that these iterations were pretty close by each other, and this is basically the same graph, but each bar is a drug in itself, and their performance across iterations was pretty similar. So we have at least some reason to believe that there might be some actual reasoning behind the ratings for the candidates across iterations. And as I've mentioned before, we have the gene ontology as our standardizing understanding of biology that drives Alzheimer's, but also which makes what makes the candidates most interesting for the repurposing. And what you can see here are those gene ontology processes that had most, or that plays the most crucial role in their in the rating. So what we see here, for example, T cell activation and phosphorylation. So we see somehow aspects of the immune system and neuroinflammation play a decisive rule for making a candidate interesting for drug repurposing or not. But now we would need to somehow get an understanding if that approach is comparable to what expert or domain experts would, how they would rate drugs for drug repurposing. We wanted to have some comparison in the literature. And here we get to one problem, and that is that pharmacology usually works within categories. So, for example, reproducible that I showed earlier is a kinase inhibitor. So if we want to have an understanding if kinase inhibitors are something that were considered interesting drug repurposing candidates by domain experts, we have different options. Of course, we could have a look at the list of drugs that got high ratings by the large language model and compare that with the ratings we get from the domain experts. But what that allows is comparing reproducible versus reproducible or propofol. That's not one of the candidates, but just as an example, against propofol, but not classes of medications or how similar medications are. So, for example, what if reproducible shows up in a high rating by the large language model and some drug repurposing candidate list curated by experts, they don't propose reproducible, but a similar kinase inhibitor. If we just do comparison list against list, then and have some binary understanding of it, then we can just compare reproducible versus reproducible, although perhaps the large language model has a similar understanding and just proposes a similar kinase inhibitor. And the interesting thing about large language models is they don't just produce text, they also provide locations of text, or they also standardize language in some way. As each word that you give to a large language model is being understood by the model within a high dimensional vector space. So in this example, we locally hosted a llama model, which works in a vector space for the embeddings of about 12,000 dimensions. So when you put a medication into that embedding space, that medication gets a vector assigned. And the interesting thing about vectors is you can calculate distances between them. So what we thought is if we want to have an understanding if that method of making use of ontology augmented prompting to the large language model for drug repurposing screening, we can use large language models not only as a language production tool, but also as a language mapping tool for understanding how close approaches of expert or domain experts are to what the model says. And this is what you can see here in 3 dimensions, which is, of course, not the 12,000 dimensions of the model, but we'll get to that later. Can we compare the understanding of experts in, for example, on PubMed with what our ontology-based prompting got to us, and that with some zero-shot prompts, such as just asking a large language model about some medication, asking it, is that an applicable drug for drug repurposing? And as I've mentioned before, this is in 3 dimensions, but Llama, for example, works in about 12,000. So how can we plot this? How can we get a human readable understanding of the relation and how close they are? And there's something that's called UMAP, and I hope you can see the video when I play it. UMAP is a dimensionality reduction technique. So what UMAP allows you to do is taking data that is in a higher dimension than the dimension you want to have an output in. And this is here shown with 3D data. We see a mammoth here on the left-hand side, which is a 3D animation. On the right-hand side, you see a 2D UMAP projection, and here you can see these different elements of the mammoth. And this is a 2D projection of the original 3D data. And this is what UMAP lets you do. And of course, you could also apply that not to get from a 3D plot to a 2D plot, but to get from a 12,000 dimension plot from the vector embeddings of the large language models to a 2D projection of how close certain medications are mapped by the model. And this is what we did here. We had a look at how do drugs that got the zero-shot prompting, got high ratings from the zero-shot prompting, compared to ontology-based prompting versus what was proposed by literature. And perhaps this plot makes it a bit more clear. We got the embedding, so this 12,000 dimension vector, and reduced its dimensionality using UMAP to get it on a 2D plot and having a look at how close are similar drugs actually to each other. What we found out is that the way these different compounds are located, or how the model puts them to each other in that high dimensional space, is actually pretty similar to how we would do it as humans when we study pharmacology. So, for example, GABAergic drugs were, after breaking down the dimensionality from 12,000 dimensional vector to 2D, actually show up very, very close to each other, same as proton pump inhibitors or Parkinson's medications. But also, for example, experimental compounds that are chemically very different, but just have these experimental names such as CYT997, which are probably not known by the large language model specifically. It was a locally hosted model with no internet access, so it could not look them up. They were also grouped together, and this is very similar to what probably a human being would do. If you don't have a specific understanding of the chemical structure of a drug such as CYT997, you would probably group them together. And this was pretty interesting because this approach also allowed us to compare how close the screening process by the large language model performed to how domain experts would do it when they publish lists on PubMed, without or by also allowing comparing similar groups. And not just comparing lists one by one, which is pretty hard to do if you have 6,000 different drugs. You would miss a lot if you only compared identical drugs and not groups. So we also did that with some lists of pharmacology textbooks to have a look if that UMAP visualization works if we just, for example, group antibiotics together, and this works pretty well, or beta blockers. So the way the large language model encodes language and pharmacology actually seems to be pretty close to how humans would go about it, which makes total sense as these are likelihood predictors. But we appreciated that we could also depict it by reducing the high dimensional space to a 2D plot and having a look at how close they would fit. So now I talked a lot about the pre-screening. These are drugs that back to this plot. These are drugs that have an impact on biological parameters. So GO here in this plot stands for gene ontology. And we do know that these gene ontology parameters actually have mathematical surrogates in the virtual brain ontology. And the virtual brain ontology, of course, is representative of our actual models. So what we believe, what this research allows us to do is providing that interoperability between the drug bank that contains pretty much all the drugs that are in the world, encoded with gene ontology, back to our virtual brain model parameters, as we can connect the pharmacological drug via its biological processes that make the drug work back to the virtual brain variable. And this is what we're currently trying to do, is basing our virtual brain simulations on this connective approach of connecting ontologies that connect pharmacology with biology, and from there to the mathematical parameters. Okay, that was my talk about the ontological prompted or enhanced drug repurposing for Alzheimer's disease, and I'm more than happy to answer any questions there might be or discussion.